Is our sound on?
Yeah.
Welcome, colleagues, and welcome to those joining us in person in the different conference rooms of the SAMRC, as well as the many colleagues joining us online. It's a wonderful delight to invite Tulio, who needs no introduction to many of you to come and give the president's lecture at the MRC. Tulio is appointed as a professor at Stellenbosch University as well as UKZN. He is the director of SIRI at Stellenbosch and CRISP at UKZN. And recently been appointed by Stellenbosch as a Pro-Vice Chancellor for Industry, Business and Foundation Partnerships. And he is going to speak to us about some of the incredible work they are doing, but also his vision for science in this country and beyond. So Tulio, over to you.
Mm-hmm. Yes, thank you very much. Yeah, it's a great pleasure to be here. Yeah, and I will just put my presentation so I can do a quick introduction. And especially to be here and to be working with the Medical Research Council. Yeah, many of you are aware, but we have a very long partnership with the MRC for the past 25 years. Across many different collaboration, yeah, including both we have been for close to 10 years. One of the flagship program of the MRC when they launched the flagship programs a few years ago was also the South African Medical Research Council that helped to fund the Network for Genomics Surveillance in South Africa. And at the moment, we have multiple grants. And one of the things that I'm most excited with the MRC is to see the opening of grants for young scientists, which many of ours got. So we are very excited about that as growing the next generation of scientists. In addition to that, both the Professor Ntobeko, he does sit in the board of SARI. Yeah, so every 6 months he come to grill us, yeah, and keep us on site, yeah, as well. Glenda Gray before as previous president, but now as chief scientific officer. So what I have to talk to you today is about how we can accelerate genomics basic surveillance for disease-causing pathogen in Africa, but not just as academic enterprise, but more also bring a lot of the innovation area. And I was mentioning at Ntobeko is a very similar lecture that I gave last week to the managing director of the World Bank, just to see how we can generate something that moves science from the lab to the industry and still have an impact. Yeah. So a very quick overview from SARI. SARI has grown quite a lot. Yeah. We are a multidisciplinary research center at Stellenbosch University that go from genomics, data science, modeling, innovation, and commercialization. We are also one of the largest genomics and training facilities in Africa. And what we do, we have a long track record of response to multiple epidemics. We also say we are pathogen agnostic. So we work a lot with HIV, TB, different hepatitis, yeah, influenza, yeah, climate disease such as chikungunya and dengue, SARS-CoV-2, and whatever come our way. Yeah, so the last two, and I'm going to present some results, is hantavirus and Ebola. Yeah, we also grow to be this very unusual center that we have multiple locations. We have 4 physical locations. Yeah, here, one next to here to the BMRI, and that's why we have probably 20 or 30 of our group members here come to see the MRC. Thanks for coming. Mm-hmm. We also funded a biotech incubator that we call the Launch Lab SARI Bio. And then we have 2 large data centers. One is SARI, which sits at the School for Data Science headquarters, and then the SACIMA, the South Africa Center for Epidemic and Modeling Analysis, which is part of SARI now. So we grow, and I'm going to give you an introduction of that. Yeah. We also grow a lot. We are over 150 now, and we have multiple research focus and areas. Yeah. So now we have multiple professors and multiple programs going from genomics, immunology, microbiome, population health and modeling, One Health, women's health, and antimicrobial resistance. And in the last year alone, we had 3 large programs. The One Health with Kalamavian. She decided that she wanted to leave the US, you wonder why, and came here to lead a program with us. Yeah. And then Joanne Passmore, that just recently left the University of Cape Town, yeah, to also run. And then one young scientist, very brilliant also, Emily, yeah, that was trained by Rob Warren, and now she's heading our antimicrobial resistance. In addition to that, we are a mix between a research and a company that I like to say. We did professionalize a lot of our support units. Yeah, we have an ISO accredited laboratory that's headed by Lavanya Singh. She's here today. We have a large grants and contract teams by Suzette Grobler, Guleta McKees and team. We also have Edwin Wilkinson, also here, the head of our bioinformatics, work very close with MRC. We have a brilliant head of data science, Julia Tegali, that's pushing AI, and then scientific support with Cheryl Bachter and a social science unit. And across that, we also have a big partnership office that both Richard Gordon work with us and Vinda Villas and I. Yeah. So now if I go to the science, yeah, one thing that we love doing is working together that we make a difference. So the Network for Genomic Surveillance was the second genomic surveillance network to be set to be specific less than a week after the UK, where we link all the main labs across the country, yeah, and that was funded both the Department of Science Innovation and the MRC. And that's quite unusual when all the universities come to work together. And as you notice, all of them with a green box. This is a NHLS or the NICD. And we did that very well from COVID. When we identify the variants, we grow to a much bigger group. And now we expanded for both different pathogens, yeah, such as HIV, TB, influenza, and so on. Yeah. In addition to that, we sit in the WHO Afro and the Africa CDC. We are one of the specialized genomics and bioinformatics centers. This means we have a mandate to support every member state. And that's quite an important mandate because we have doing sequence now for dozens and dozens of African countries, but we really train like most of the countries. Yeah, we're hitting a milestone of 1,000 fellows from 50 African countries. And included here, even in this room. Yeah, how many fellows we have in this room here? Yeah, so 10. Yeah, we have 10 here. And that's exciting because the idea is that they get very advanced training and they go back to the country to respond to epidemic. Yeah. So this lecture is going to have 3 pillars. Yeah, the first one, we're going to invert the way that we normally present. We're going to talk first about not science, but how do we work with big industry, but also how we create the industry that we need to develop local biotechnology and pharmaceutical manufacturing capacity. And then I'm going to show, when finish this pillar, a small video. And Dr. Richard Gordon, that's here with me, is one of the main actors on that video.
We're doing autographs at the end.
And then I'm going to move on something that people don't know very well, but it is the bulk of us are data scientists. Yeah, how we can use not only data science, but AI to main epidemics, to do data set at scale. And a lot of the data sets is not genomic data. We are talking about satellite data, mobile data that we do that to understand. And then I'm going to finish how we can really pivot very quick with example of both hantavirus and Ebola. Yeah. So if we start the first one, we're going to go working with industries. So we are very excited that we are a founding member of the mRNA WHO consortium, which is really working with the MRC. And last week we host the main annual meeting at the BMRI. Yeah. So here is a great example how we went for one of the first time, if not the first time ever in Africa, from identifying a pathogen, sequencing the genome, identifying the genome, feeding back that to then production of mRNA vaccine in Africa. So it's going end to end with Afrigen now taken to preclinical trial with a large award from CEPI. Yeah. And then we also do that with also big companies. Yeah, this one I'm very excited. Yeah, because maybe a game change. On the left there, I show a graphic. On the blue, it is Asia, and that artemisinin resistance. Artemisinin is the main drug used to treat malaria. That in Asia, and that the y-axis go to 100% basic. Almost every sample of falciparum is fully resistant to artemisinin, and that's entering Africa on the bottom very strong and growing very quick. And unfortunately, what you see in the last 2 years is increasing not only artemisinin drug resistance, but also case in Africa. We partner with the GSK, and we just got funded a large award called KUMAL. That's a 33 million euros award from the EDCTP, where GSK put half of the money, which is to evaluate a new drug called GSK 3772701, yeah, which is a potent, fast-acting falciparum wholesale inhibitors with a novel mode of action that is active to sensitive and drug-resistant parasite. And we're going to be performing all the DNA sequence for the malaria parasite of this new compound. And we are very excited, fingers crossed, because more than ever, we need a strong new drug, especially if this trial succeeds. Yeah. And then what we decide that's not only work with big industry, but try to advance the industry of the future. So what we did in collaboration with the Launch Lab and Stellenbosch University, we built a biotech incubator, yeah, where we refurbished 300 square meters of lab, but straight away to make that ISO accredited. This is 13485. This means anything produced here can be used for the diagnostic and the medical device. We launched this biotechnology incubator and to bridge a critical infrastructure gap between academic research and regulated biotech offering. And what we did that was quite exciting, we used the space of a company that they have moved out, the Cube space, that's becoming the first unicorn of Stellenbosch. So that's excitement that people also want to become a unicorn. And a unicorn in innovation is not that funny horse. Yeah, it's something value of $1 billion. Yeah. And that is a piece that came on the Stellenbosch video that really highlighting that believe that a quiet revolution has been unfolded in Stellenbosch University campus, not in lecture halls or live courtyard, but inside the glass walled co-working space and labs, where the students, research, and entrepreneurs build the company from scratch. And that's the work of the Launch Lab and Innovus. At the moment, they have 50 companies being incubated in Stellenbosch University and over 10 biotech companies, just highlighting that's beginning to happen there. One of the companies that it's part of the SAMRC family, or at least we have a percentage shares on that. It's a company called Fluoro Biotech, yeah, which obtained the ISO 13485 certification. And that's also a success story because that also linked to the MRC because they received many of the MRC trainees, not Richard? Yeah.
So this was spun out of the mRNA Hub program, making the enzymes. So, and then they took some of the trainees that were done at BRIP, and now most of their staff in that picture, all of them came through BRIP last year and the year before.
Yeah, and that's great. They grow from 2 to over 10 full-time employees, and they have signed contracts with multiple companies now for the first time to be able to produce enzymes in South Africa for mRNA. So just to highlight that if we are to decrease the price of the vaccines, what's quite important, otherwise our government will not procure, yeah, we also need to start producing the enzymes here, yeah. And that's a company that we are very proud to that. And then we thought that not only on the companies in Stellenbosch, but that how we can do that more with a continental scale. We did a partnership with the Open Startup International and the MIT Executive Education Program, yeah, and twice, 2025 and 2026, sorry, there is a typo in my slide there. we host what you call acceleration program, that we put a call to all Africa. Last one, 300 companies apply. It is companies to grow between like a few employees to maybe 50 or 100. We select 10 companies, they stay 1 year in mentorship, and they come for doing a very intense program here. And that's very exciting to see this partnership and bringing a lot of the startups, the top ones here, to be accelerated. Yeah. And then we had an idea end of last year, one of these ideas that just happened by what, Richard? Just by drinking wine and running.
Serendipity.
Yeah. Serendipity. Yeah. So we have here just to show 2 people that can visit us, and it's going to be clear that's Dorothy Shu. She's the head of partnership of Google DeepMind, and that is Janella. He's the head of partnership of OpenAI. You're going to see that we are working a lot on AI. They came to visit us. We have a partnership with them, and they also angel investors. And then they come in, our startup owners. Yeah, in the photo they took them for a long run on the top of the mountains at Cape Epic. And then when they come, I joke, instead of running, I went drinking wine. And then I joked to them that I would bring them in the hottest time of the year to sweat here and invest in companies. And then came the idea of doing the Sweat Africa on the back of the program, the BRAIN program with the MIT. Why that? We are already flying with Open Startup 10, 70 people, 10 top companies. They say, why we don't start a big program? And that's what we did. Yeah, that in the end was quite exciting. Yeah, we tried to cover the number of participants at 250. We end up with 500, and we receive 150 startups from 18 African countries. And some of them big ones, other ones small ones, but was the first time that we bring not only all these startups to one large event, but also a lot of the investors. So just if I can give a zoom here. So we had startups on different areas: agri-tech, AI, biotech, deep tech, fintech, green tech, hard tech, and health tech. We put a competition. We had 80 startups competing for the prize that was on hard techs, making hardware and biotech. And one of them on the big prize, yeah, called Urubo Biotech, yeah. But more important, what we did is to bring also the big investors together. So what we did, and the MRC was there, the funders, TIA was there, but also some of the large VC funds in Africa, like AfriInvest, AfriGrow, yeah, took UTF and the biggest investment bank in Germany, KfW, to come together with the startups and then to start investing on them. And we estimate over R1 billion could be invested on that. And we know already about dozens of deals coming that, yeah. So what I'm gonna do now to you, if work and should work, because we test with the IT, I'm gonna play you 2 minutes of the video of the Sweat Africa, so you can understand what was all about. We need the sound here.
Wellcome has committed to spend £16 billion across 10 years, and we are calling on you.
Okay. Sound. Yeah. Sorry? You say that it must go through the crystal. Can't put out the sound. Yes, but we did that before. Yes. Okay. Yeah. Please tell me if you can hear online. Yeah.
Yes. Yes.
Wellcome has committed to spend £16 billion across 10 years, and we are calling on you.
You cannot hear online.
On the continent to come spend that money to invest in the areas and embedded in the contexts that are facing these health challenges.
I think you only need to be here for the last 2 days to realize that there is innovation all over the place in Africa. However, it's in pockets, it's in silos. So the whole idea behind this meeting is to get young entrepreneurs together with funders and start building ecosystems. Because until we start building ecosystems working horizontally between countries, we're not really going to create sustainable livelihoods and businesses for people in Africa. This is the first meeting of its kind, getting ecosystems from different countries all over Africa together.
You need the entrepreneurs, you need the funders, you need the university because they bring the important research. So all solutions informed by data, and then we can solve all the problems. In Africa today, every year for a decade, we've been seeing scientists doing amazing work in the lab, but when going to the market, we get stuck. Unfortunately, knowledge that doesn't move does not create economic growth, does not create general system where everyone can grow together and prosper. So actually what we are doing at Open Startup is creating the next generation of scientists that are creating the next jobs, that are solving the problem of the continent, using technology that is being developed locally. And we are trying to do this really connecting the north of Africa up to Cape Town here, making sure that there is a corridor where science is on the move, where the science is out of the lab, out of the university, not only being cited, but really being used to develop products, services for a better society.
I'm Billy Olojade. We just finished a panel that was exploring how ideas go from the mind into the lab and out in the real world. And how do those ideas get funded and by who? This is a very powerful way to show our young entrepreneurs how to start their journeys or to accelerate their journeys. And all of this matters because these are the things that will make Africa progress.
So as that.
Then, yeah. So as you see, was a very exciting event. Yeah, not only excitement, but we saw a lot of deals being made by that event. We have the new dates for next year. Yeah, so it's going to be 16 and 17 of March. Yeah. Please pencil in your calendar red, not tobacco, yeah. And we're gonna probably double or triple the size. So next year we're expecting 300 to 400 or 500 startups. But the important thing is to get the right ratios of one investor per 3 startups. Yeah, and quite important, the SWET Africa. Every single big university in South Africa signed. Again, another distinct UCT, WITS, University of the Free State, UKZN, Pretoria, and all the ecosystem builders. And that's what we love doing, is break these boundaries and make people working together. Yeah. I will change a little bit topic, but now to talk about how we use big data at scale, and how we are also using AI to deal with the main epidemics in South Africa and Africa, and also in another big pillar that a lot of the startups that we had before is to use what you call deep tech, also to train people that can enter also the market of AI. So SAMRC has a relatively big data science infrastructure. People don't, a lot of times they think that we are a lab group, to be honest. Yeah, Lucius, how many people work in our lab? 6, and we are 150. Yeah, so a lot of our people, it is data scientists. Yeah, why only 6 in the lab? Not because they work day and night, it's because mostly robotic. Yeah, and so what we did is to invest a lot on data scientists, software engineers, and AI scientists. They sit both at the headquarters of the School for Data Science and Computational Things at SACIMA. And then we did a big program with the African Institute for Mathematical Science and the Google DeepMind that called the AI for Science program. Yeah. And that's what we are doing. We are using quite a lot of AI now, to really start not only working backwards, but start to predict, where big outbreaks will happen. Yeah. And what we're doing we're doing this very, very advanced multi-scale convolutional density ratio estimation, look quite complicated, but maybe what we do, we get this very big layer of data sets, of ecological data set, satellite data set, climate data set, pathogen data set, and we can calculate where is like outbreaks be happening. So I'm just gonna zoom here. Here is for dengue, dengue, we have calculated that one of the big areas that in Africa that would expand large would be this belt in West Africa, where we expected 6 million people in 128,000 square kilometers to be at risk. And then what we did is then to work with our fellows that we did to prepare them. So here is one of this country. Here is Togo, just to ground you. Here's the capital Lomé, here the Atlantic Ocean. And what we did is to work to then not only to do the genomic surveillance, but to have diagnostics there. And they end up uncovering in 2024 one of the largest outbreak of Togo ever. These would be samples that normally would be negative from malaria, that people wouldn't test, but when they test, they uncover this very relatively large outbreak, or the biggest of the history of Togo, of dengue. And then when we sequence them, and they come here to sequence in our lab, what we found is this very unusual scenario, Just to ground you, here is a phylogenetic tree of dengue serotype 1, genotype 3, lineage A. And here in blue was a big outbreak that happened in 2019 in Tanzania, And that is a strain that caused a very large outbreak in Tanzania and had disappeared. And all of a sudden is re-emerge with the biggest outbreak of the history of Togo, Burkina Faso, and Cote d'Ivoire, with at least 5 introductions, just highlighting that probably we found a new corridor of what you call crypt transmission. And in normal English, is that intermediate locations that the virus could have gone, but is undetected. So now we are uncovering this East-West Africa corridor of diseases, which is for dengue, for example. I'm going to give you another example of another virus that is chikungunya. Chikungunya is a virus that has been identified in 1952, again in Tanzania. Poor Tanzania is not, they just have good systems, And that is a virus that was causing only normal small outbreaks until 2005. One key mutation emerged, one key mutation in the receptor of that virus that allow better transmission, not between the virus and humans, but between the virus and a mosquito called Aedes albopicus. That's a mosquito that live at higher altitude or colder temperature. And then the virus from that mutation invade Asia. It caused large outbreaks in Europe, invaded the Micronesia, and then it's introduced in the Caribbean and caused very, very large outbreaks in Central America and South America. This virus, it is a very problematic virus, not only in the mortality and morbidity. People get so sick that they bend. That's the chikungunya word for bending the floor when you're crying. But many people that get infected with chikungunya will suffer forever a lot of pain in the body. A lot of people cannot go back to manual labor. Unfortunately, that same mutation happened again. So just to ground you, here is the mutation, the A226V, that happened in 2025, that caused a big outbreak. That's what is spread. A completely new emergence of this mutation happened. First detected in Central Africa. All these green sequences are produced by the US Army. So defense really worry about this virus because it can invalidate a lot of your army battalions. And then it entered into August 2024 in Reunion. A massive outbreak happened in Reunion. We're talking about dozens of thousands of cases epidemiological week, and then you knew very quick that once entering Reunion would spread widely, and especially entering Mauritius. And then we worked with both Dr. Vanessa Ramuth, that is from the Minister of Health of Mauritius, and not doctor anymore. Now Julia is a professor. She just has been promoted, Professor Julia Tegali, head of our data science, of not only track that, so we send reagents to Mauritius, but then the Mauritius Minister of Health start taking that serious, and they start controlling the mosquito population before the outbreak happen, and then tracking real time, and they end up with a much lower outbreak size now in the dozens of cases per week, not in the dozens of thousands.
What percentage of Reunion got chikungunya?
They estimate around 70–80% of people
of everyone. That's insane.
And much less. And that's again, quick response, preparation, and that was quite important, not only on the science, but to respond to that. And this new variant of chikungunya has potential for global spread and biosecurity. That was covered in science. After that happened, that this is really expect to spread beyond the Indian Oceans. And we know that problematic. China had the largest outbreak of chikungunya a few months ago. And unfortunately, the vaccine that they tried was discontinuous, because side effects. So they also don't have an efficient vaccine, And I can talk for another 5 hours if you guys want. So because the same as the last two, I highlighted the dengue in Togo and in Mauritius of chikungunya. We have responded to over 20 outbreaks in 17 countries in the last 2 years by sequencing genome, producing reagents, advanced data analysis, and capacity building, And then what we did is then to do that not only from the lab, but also from using that from also AI and also with dashboards. That's a dashboard that we have produced. We have done first for SARS, then we adapt for dengue and chikungunya and Zika, and that's been very widely used to follow that in real time. And then last week, we also adapted to hepatitis B and C, and we got a very nice publication. So just advancing that. So before I finalize, I will play you another short video because sometimes it's easier to see than to what's happened. That is the program that we have with AIMS and Google DeepMind. That's one of our fellows. That's the poster boy, and it's just a minute and 20 seconds. So let's see that. It's starting the video.
I grew in Yaoundé in Cameroon in a neighborhood called Ikuni. Research focuses on dengue virus and arbovirus in general, which are understudied in Africa. AI is becoming better at identifying diseases and showing us how climate change impacts the spread of dengue virus, specifically in the West African region. I want to use machine learning to help identify these kinds of diseases and improve tracking in Cameroon. I think for AI to help as many people as possible, it needs to be built by a diverse group of people. That's why making sure more people have access to higher AI education is really important. Best part of AIMS was connecting with people from all over the world, like AI experts and mentors. And the cutting edge techniques I wouldn't have been able to get anywhere else.
So what we found it is that it's so important, to bring these new technologies. So as I highlight, our lab, it is very automated, fully robotic, or mostly robotic. We are investing a lot on artificial intelligence. This year, we graduate already the 3rd class of the AI for Science program with AIMS and Google DeepMind. We have incorporated many of them to do PhD with us, because we need to bring these new technologies if you to do that in real time. So just to finalize, I'm just going to give 2 examples how we bring all that together to real-time data system that can support rapid detection coordination to high consequence pathogens. And I'll mention the fantastic work that our teams are doing in the 2 recent outbreaks on hantavirus and Ebola. And the main take home here is that there is much more than just a sample and a genome. you have to really bring a lot of the other information together to understand that, and a lot of them with advanced data science and AI. Yeah. So what we do when we see a new outbreak, we put what we call a rapid response team. Yeah, where we get multiple scientists, and that's a lot of our top scientists work full time. The first thing that we do, we create a big folder, we compile all main scientific publications, and we read, and read. So in the hantavirus in the first week, I think in the first 3 days, I probably read like 50 papers. And that's just to try to highlight to our people that you need depth of scientific information, especially for new outbreaks. Yeah. And then we have the team, after reading a lot of this reading, a lot of time involved even a different language. Here's, for example, the bulletin in Spanish of Argentina. Yeah, that happened. And then we had the whole team, a big team, doing the large data analysis with both Julia Tegali leading that team, yeah, Edwin Wilkinson in the bioinformatics, and then Carla Mavians in the One Health. Then we attend many of the WHO and Africa CDC. I'm part of a lot of the closed briefings because I sit in the technical virus evolution of the WHO and the Africa CDC in the strategic advisory on genomics, but we also attend a lot of open drifts. We do not only writing articles, we are getting quite a few big scientific articles, but we also focus a lot on scientific communication. It's quite important because a lot of the time you have to also communicate to your government and to the public. Yeah. So just understanding what our teams have done on hantavirus, that's bringing these very large ecological data sets that we have. This is really rich data sets that we have for the whole world. Yeah. And then bringing that in both AI, but also traditional epidemiological modeling. On the latter is when you combine now the presence of the virus and the presence of the rodent. These virus are normally the main habitat, the main host is the rodents, and then you start getting a quite idea where that can happen. Yeah, one thing that you start mapping from satellite data distance to waste sites. Why that? We know that waste sites have a lot of rodents. Yeah, and then we bring like temperature, temperature anomaly, and we can start seeing with these models what's begin to explain not only the geographic, the distance to potential sources, but also what's happened in that year. And then we start developing these models for ecological susceptibilities, for, in that case, the virus of hantavirus called the antivirus. And then we started bringing big layer of big data, such as population density, build-up intensity. That's where the big cities are in South America. Tourism intensity, we know that's quite important because a lot of people get the hantavirus from tourism activities, especially when they go to the remote areas. And then we begin to link that and track to the previous spillover susceptibility. And then it became quite clear where it is happened over the years. We calculate then the number of people that are exposed per year, and it has been increased. 2025, we talk about 52 million people that could be exposed to that, but only we had 60 cases in Argentina. So we completely can already understand that this virus, by the exposure, is difficult to be transmitted. It's very niche. It has to be in close proximity. And then we start understanding like where is like to do that. Then when we map the public data, the genomic data, it's become also quite clear. It's here the 2 different strains or the main strain that was found, that was initially sequenced, that was made public. I'm just gonna zoom here, by the Netherlands and Switzerland. Yeah. And then again, when you look about that, it is basic no genetic diversity. This means that they are very identical, with strains close to Argentina. So we know that this virus has not evolved like we saw with chikungunya or dengue. Yeah. And it's also very close to a strain that exists in Chile. Yeah. Not surprised when you look at the geographic distance, despite they being in different countries, they are really close to each other. They're just by the border. Yeah. Mm-hmm. So now we know like the parameters, the strain, the evolution, and so on. and that we can use that information for both scientific response, but also to public science of communication, which I find that's even more important these days as people, because a lot of time governments will take the actions and individuals and countries, not just by scientific paper, will take weeks to come, but how it's transmitted. So we got multiple pieces in the New York Times, in Al Jazeera, BBC, that just to communicate about that. And our communication was quite accurate. I was going to show you another short video, but I will pass that. That's the guy, the main host from Al Jazeera. Al Jazeera probably is now the TV that have the highest number of viewers in the world. I wonder why. Yeah. and sometimes what you're gonna do is that these have to be communicated very calmly, because people will push you. Yeah, everyone wanted this big panic about hantavirus, and was clear that was no need to panic. This virus exists for 50 years. Yeah, we know what it is. We know people expose it. It's no mutation, and that was quite important. Yeah. But I will spare you of that short video. I can send you after. And then we are doing the same with Ebola tracking. Yeah, all the cases, yeah, the locations. But then again, with the big data team, you start looking about, yeah, the ecological niche. That's where it's likely, and what's associated with deforestation, human density, mining proximity. Yeah, why do you think that we use the other one? We use waste, yeah, like waste sites. Why do you think that we are using mining here for Ebola?
In the cave, in the mines, the air spreads.
The air spreads? No, Ebola is not air transmitted. What's the natural reservoir of Ebola virus?
Bats.
Bats.
Yeah.
And bats tend to be a lot of bats in caves, and caves are exposed by mining. So that area is a high mining area. Yeah, but quite. Sorry, let's go back. Yeah. So here's the area that have emerged. And then one of the things that came very strong is the conflict exposure. So that's exactly the area. It has the perfect things. Yeah, it has a high spillover chance. Yeah, for that new virus when you mix both the ecological, the intensity, the urbanization, the location, and so on. So unfortunately, it's not surprised that they have emerged there. Yeah, and it's a very difficult area to deal with. At the moment, we know that a lot of the teams have great difficulty to identify the patients because big conflict and so on. But what we want is to use this real data in real time to do that. So one of the things that we saw that mobility was very restricted. It's very difficult to live to that area. Yeah, high conflict, very far away. And then again, so we can communicate. We communicate that both scientifically, we finished the results, but that was a piece that I'm cited in the New York Times again. What was the risk that to happen in Kinshasa? And was quite clear that the mobility is very restricted. So we have a very low risk for a big introduction. Yeah. And that would be much better to control that at close of the site, especially as they close the main airport at Ituri. Yeah. Then to finish, what we are trying to do here, a lot of this work. So for example, on the Ebola, now we are working with one of our fellows at Uganda to see how we can support them in the same way as we did with dengue and chikungunya, with reagents, with diagnostics, with data intelligence. Yeah. And a lot of this work, why we can participate and respond to a lot of these outbreaks everywhere, is just because we have an army of fellows. Yeah, we have 855 fellows from 48 African countries. Yeah, that we have trained in advanced genomic surveillance. We have here multiple fellows. I think that we have here that we are training on computational biology and other rare skill. That when they go back to the country, we can then work together to respond to that. So one of our fellows in Uganda, I don't know if you want to bring more information, Richard. You just had a session with funders. Yeah.
And Tobacco was on the call. So one of the African Stars fellows, Kabuga Joel, yeah, he his day job is he's works for the Ugandan Ministry of Health in their surveillance labs. I think he's a group leader. So he last week Monday got in touch with the Ugandan government, and then he presented to 20 odd funders on a call last Thursday to see who can support the reagents and shipping, because a lot of funders are giving money, but they're giving it to WHO, giving it to Africa CDC, giving it to CEPI, and then it takes like 6 weeks to 10 weeks for that actually to get anywhere. So the idea is that hopefully we can send reagents, see if we can tap the MRC president up for the slush fund as well to be able to respond quickly. So so that's how you use the network. You know, the previous examples you saw, like dengue in Togo, that was a fellow who came here from Togo who got trained and then either sent samples here or we sent reagents there to respond.
Yeah, in Mauritius and Burkina Faso and what we found here to the president of the MRC, so my pitching for his LASH fund, is that we found that to be extremely cost efficient. Why that? Because Dr. Lavanya Singh run our lab. We have this massive genomics lab that's using a lot of these reagents as high throughput. So first, we can procure much cheaper. Second, what we do, we take part of our reagents to send to them. So the chance of expiring is also very, very short, because a lot of time when we send reagents to a country, we need reagents that come from 5, 6, 10 different companies. And even if they get the money there, they would pay much expensive because they're not buying bulk, but also would take them to source from 10 different companies. Will take weeks and weeks. So we find not only rapid, but also very cost efficient.
So I was in part of a malaria sequencing project last year, and we spoke to 4 sites in Africa and said, what's the average time to get your reagents? Exactly the same thing. Well, there's like 6 or 10 different things you need. The shortest we got was 12 weeks.
Yeah. So just to conclude, So SARI, it's more and more a center of excellence. We are already both recognizing South Africa with the National Genomics Surveillance, with the MRC, but also in Africa with the WHO AFRO and the Africa CDC. We are beginning to have a good track record in genomics, but not only on that, on epidemic response and capacity building. And that's why we find so important all this fellowship, because then we have fellows everywhere in Africa that can respond. We have a very advanced data analysis expertise, which has been expanded to include AI. It's absolute phenomenal what is happening both on the bioinformatics, data science, and One Health. I cannot make juice to all the work of Huria Tegali, so I just show a tiny little thing, and her team, they are doing phenomenal work. And we are really well placed to provide an ecosystem to expand biotechnology incubation and acceleration. And the idea is not only at SARI and Stellenbosch, we did that with the MRC, with TIA, with all the main university and ecosystem builders. Also to enhance data analysis to understand epidemics, outbreaks, and spillovers. And just to enhance the biobanking for surveillance. So this means not only receiving samples, but supporting many countries with reagents. And we also see that the MRC even copy us, and now they have their own magazine. So you copy the gem. When did we start that?
Well, ours has been running for many, many years.
I didn't feel like as a magazine. I saw as a newsletter. So we have a very good scientific magazine, which all of you, and we don't mind if people copy. Normally, it's a good sign. What's the name of your magazine? Bopilo. So we have the gem, we have Bopilo. It's great because science communication is so important, especially because the people that take decisions, they're not going to go and read your Nature paper or your Lancet. They will get from science communication. And everyone can scan, read the gem. At this new version, we have some of our new MRC young fellows that got PhD fellowships, but also a grant on West Nile. So we are highlighting the great work of the
fantastic output, the gem. I really enjoy reading it.
Fantastic. Thank you. And then just to really, I really have to thank the funders. Yeah, I put the MRC on the top there. No, but it's not a joke because despite a small percentage of our funding come from national funding, they are very important because show like support, national support, which allow us to leverage. It's a very difficult time now for funding. So what we do is to do a lot of that. And then to finish, I also want to invite the MRC to also come visit us. So now on the Thursday, 11th of June, what we did, we created a new lecture series, what you call the Pro Vice Chancellor Open Lectures. That's to open Stellenbosch with the main objective to work the university external people to break barriers and open communication, collaborations, even internally within faculties and research center, but externally, yeah. So we are running that under the Provis C. Last week we had a beautiful one with on the HIV, social science and costing with the Faculty of Economic. And this Thursday, just here next door. So please come. We also offer you coffee and nice tea. What we're gonna have, we're gonna have another external presenter. Yeah, his name is Sudep Duga. He's gonna talk about mitochondria from biogenes for pharmacology, but quite important. Yeah. So, for example, he got a big award, including the USA National Inventor of the Year Award. And he had founded and helped build several pharmaceutical and biotechnology companies, yeah, including Advanting Pharma, Sphera Pharma, Imperial Bio, and Island Therapeutics. And that's the kind of things that we want. And that's the Provis lecture put in partnership with the Africa Center for Therapeutics and Innovation, yeah. And we're going to be hosting here at the BMRI, 200 meters from you. And not only the MRC people is welcome to come there in person, but also it's going to be online. So thank you.
Well, thank you very much, Tulio. That was a fantastic tour de force. And I want to congratulate you and your colleagues at SIRI for the phenomenal work you are doing. And I'm sure there'll be lots of questions both in this room and online. But I'd like to start by asking you a question. I think that's probably one of the most impactful aspects of the work that you are doing is the capacity development on the African continent. And the question I want to ask is, how do you actually measure the impact of your capacity development efforts?
Okay, so yes, there is many ways to measure the impact. Yeah, but let me tell you, yeah, sometimes is not difficult to measure. So, for example, I can give you a few examples. Yeah, now the scientific head and all the head of the labs of Botswana. Yeah, Sikolile Moyo was a PhD student of mine. Now he's heading that. The whole head of the genomics and innovation program in Zimbabwe, Justin Manassa, was a PhD student of us. Yeah, the head of the Instituto Nacional de Saúde, the National Institute of Health of Mozambique. Yeah, Nalia. Yeah, Ismael. Yeah, all the genomics. She also did a PhD with us. Now the head of the National Public Health Institute of Cameroon. Yeah, Renee Osomba. Was a fellow of us. No pressure for the but that's something that really gets to us when we see our fellows not only having their own impact scientifically, but taking big leadership positions. Yeah. I would not be surprised in the next 10 years if most of the head of institutes and many of the ministers of health were fellows of us. Yeah. So even from this new cohort, and we just wrote what we call 7 impact stories. Yeah, that's with the Mastercard Foundation. Yeah. And it's just amazing what some of them are doing. I don't know if you want to highlight 1 or 2 of the recent impact stories.
Sure. I mean, obviously, the most recent one is the Uganda response. But it's been fascinating to see how the evolution of the fellows, giving them non-scientific projects, more entrepreneurial projects, and how people just find their niche very quickly. So now some of the fellows now, they're working on 4 different projects, and they've got a target of each submitting 3 different grant applications per project in the 6-month window. So you're moving it from just working on now engaging with funders and seeing the progression of pitching to funders from day 1 and day 2. I think probably the easiest example to see is a few years behind is you look at the mRNA Hub program. There's literally been 3 companies that have spun out of that program. You know, Afrigen was there, but there's the Fluorobiotech, there is the BITS mRNA program, and the novel lipid platform. So how different scientists find their different careers. There's lots of stories about people getting jobs and promotions that can be part of it, but and to back and answer your question, it's not in patents and papers. It's seeing the impact on the ecosystem.
Yeah, but also I must also say that probably not just because our fellows are really, really brilliant, because they come from, for example, how many of the bioinformatics, computational biology fellows I have here in the room? If you can put your hand up. Okay. So we have 1, 2, 3, 4, 5. Just a quick note about 1,300 applicants, we select 12.
Wow.
So even if we screw it, then they're probably going to succeed alone, because they but very quick, can you tell quick your name and where you come from? And okay. Yeah, first woman. Yeah.
I'm Beatrice Mbamba. I'm from Malawi. I'm Tato Penyo, and I'm from Botswana.
I'm Wadafiano from Ghana. I'm Donald Abbott from Kenya.
Moyulu Afalowo from Nigeria.
And we have so they came from a big selection. We try to provide them not only technical training, they are all under Edwin Wilkinson, our head of bioinformatics. Is he treating you guys well? Very well. But then what we did, we do things that is kind of that what you call the strong homework. So the bioinformatics and computational biology took us 2 years, but we reached a structured masters. So they have 9 months that was approved by SAQA, not SAQA. SAQA. Yeah, took 2 years process, but now they have 9 months of training, of programming in different areas. And then after they go to do a research program. So the AI program that AIMS have put, that we are one of the main anchor partners, they have again 9 months of very extreme AI training by the best people in the world, people from MIT, Google, Microsoft, Oxford, and that. Our African Stars fellows, what we did, we give them we give them 18 weeks of very intense training that involve a lot of time, grants management, financial management, leadership, emotional intelligence, how to talk to the media, how to deal with rejection is a difficult one. It is. With a scientist. It is because not everything that you're gonna do is gonna work, and you have to learn to bounce back. So we have many ways. The ways that we track the impact, it is employability if they are advancing their career, and now more and more if they are generating not only papers or patents, but if they're creating companies or if they're helping the country to advance. Another thing that we don't do a brain drain. So 99% of our fellows go back to the country. And that's why it's so important with the MRC/fund to support them when they see something there.
That's wonderful. Let's see if there are other questions for you in this room, colleagues online.
That's it. Online, you are welcome to type your question or ask.
Maybe I can ask you
Yeah.
another question, Tulio. So you're probably one of the most recognizable scientific faces in this country. And I wanted to ask you to just chat a little bit about your approach to scientific communication, given the receding trust in science everywhere.
Okay, maybe I should play the last little video of Al Jazeera, because you're gonna see that, yeah, because that's quite good, because and it's not because me. I was no, because it is very important, yeah. Science communication do not happen without strong science information, yeah. So what we do, and I just gonna play to you so you can see how much this guy push and how much you can get in trouble if you don't have a lot of the information. So I had 10 of my team members generating information in real time. We read all the scientific papers, yeah. And then you have main science communication, it's science information, which you cannot do alone. You need a whole team behind you. So let's just see. He's the main anchor of Al Jazeera, so he's quite senior. You're gonna see how much he pushed me.
De Oliveira is the director of the Centre for Academic Response and Innovation at Stellenbosch University. Joins us now from Cape Town in South Africa. Thank you so much for joining us, Tulio. Now, everybody says it's very difficult to get this particular disease, this virus anyway, but facts on this ship would seem to prove otherwise.
Yeah. Thank you for having me. Yeah, it is very difficult to get that disease. Yeah, especially when we call human-to-human transmission. Yeah. But what's probably happened in the ship, yeah, is limited transmission. We don't expect all the passengers to be infected. And what you have to remember is that these passengers then spend over a month together in close proximity, yeah, in a ship sailing through very cold areas. So a lot of time inside. So what we know is that this virus, it's only transmitted by very close contact. So we are not surprised that the first 2 infections that people end up dying were a husband and a wife, which the wife ended up unfortunately dying when she was here in South Africa.
Tulio, so I mean, if you look at it, one German woman died a day after flying to the Netherlands, and a flight attendant admitted to hospital showing symptoms as well. So it's not just close proximity for a long period of time. That would have been very brief.
I think that you have to be careful at the moment to assume that the flight attendants that show symptoms, it is infected with the hantavirus, because a lot of the initial symptoms of hantavirus are common symptoms such as gastrointestinal symptoms, GI symptoms, or fever or headache. So I think that we should be a little bit cautious to assume that is hantavirus until the diagnostic results that are being done at the moment. We have reasons to understand that in the Netherlands, some of the most advanced teams on public health that are looking at that. So I think that we have to be careful to assume that that's a hantavirus before the final diagnosis.
Tulio, I mean, you're a global expert on this particular virus. How confident are you that this is not going to result in some kind of pandemic?
Okay, so this virus is not new. Yeah, it was discovered in 1976, 50 years ago. Yeah, it first spread quite widely in Asia and in Europe. Yeah, and then have entered Latin America and South America. Yeah, Argentina, the country that it is at the moment, the epicenter, or that have the highest number of cases of hantavirus in South America, we are talking about 60 cases a year. Yeah, so, and most of them acquired direct from animals with very limited chains of infections. Yeah. So if this virus were to cause a pandemic, yeah, we would already see a large number of infections happening in Argentina and in other countries, but we don't see that. And all the biology, all the science that we know about the virus, hantavirus, and even that specific strain, the Andes virus, which it is transmission between humans is limited.
The fact.
So the question I have to you is, did you know before that if the airline stewardess wasn't hunted?
It was very unlikely to be hunted, and she tested negative.
She did. That's what I'm asking. Did you know?
I didn't, but that's what can you see? But that's what they say. You have to be careful to assume, you know. So and he will push more. He will push now on the evolution of the virus that the virus having. So to talk to the media, so you need a lot of information. A lot of the information on these emergent pathogens is counterintuitive. Everyone assumes it's a boat that would be a respiratory pathogen. So you don't assume, you read and read. One of the papers that was very important for me from the hantavirus is a very, very technical paper when they outgrow the virus in various cells, and they outgrown the virus in different components, yeah. And then on the nasopharyngeal, and it has almost no virus. So it's going to be clear, quite strong, that that virus would not circulate a lot in the respiratory. In the blood, it was a lot, so it's very unlikely. So a lot of the time, the information that one have to take is not from the, doesn't help just to read the media, yeah, and try to learn from the media. You have to go back to the scientific, yeah. With our teams, yeah, we are tracking the virus, yeah.
It's fantastic. I thought I saw a hand go up online.
Len. His name is Len Henmark can speak.
Aline, please go ahead.
Thank you very much. And thank you, Tulio, for an amazing description of the many strands of your work. I'd like to follow up on your network of fellows and ask, what are the kinds of challenges that the fellows have faced? Or could I say, what are the most common kinds of challenges that the fellows have faced in going back into their own environments and facing, you know, the very real limitations, I think, in many environments. So in addition to funding, you mentioned funding, but what are other kinds of challenges which your fellows have been facing? Thank you so much.
Yeah, yeah. So yeah, a few times it's not easy to go back. Yeah. And that's why we still try to support the fellows with both shipping reagents or getting samples or getting them back or working in collaboration, because we find that we can help them on that. Yeah. Some of the fellows also decide to change work. Yeah. So for example, they get promoted or they apply. But one thing that we try to teach them, it's on how to do a lot of applications. Yeah. So to do PhDs or postdocs if they want, or to apply for new jobs. Yeah. And some like that. So, but to the best of our knowledge, in our last survey, yeah, over 90% of them were still back in the country. Yeah. So we also try to encourage them to go back to the country. Yeah. And yeah. So that would be, I think that as some of the difficulties is that is less access to resources, yeah, and yeah, then to go back. But yeah, what we're gonna be doing, Ellen, yeah, we're gonna be hosting this year the first alumni program. So we're gonna start getting alumni that we can get in contact on them to try to record that, yeah. And two groups of fellows have wrote a paper highlighting the experience and how they put that back in the country. We are also happy to share that. Yeah.
Tulio, maybe I can ask a final question. Why specifically the Bundibugyo strain rather than the more common strains of Ebola this time around?
So I think that it is very clear, and that's not our work. It's just analyzing the work that was published on the phylogenetics is a new emergent. Okay, so it's again the virus that live in the environment and cause outbreak in humans. So because that it has to be either a new strain of an unknown previous strain or a new one like the Bundibugyo. So that's what's happened. Yeah. And the last time that they saw emergence of that virus was around 10 years ago. Yeah. So, and that again, it is quite funny because can you see by being a evolutionary biology and working with epidemics for decades, yeah, that's the common process. Yes, normally is a new emergence of strain, and we know, like from imagine even here in South Africa with HIV, it probably took us decades to really accept that was emergent from a virus from a chimpanzee that have entered. You remember the confusion in the past? Yeah, TB we know that emergent from bovines. Yeah, influenza. We know that every year come from either avian or pigs, and now we open now opportunity to come from cows and other mammals or cats. Yeah, and then so we know that Ebola normally comes from bats, hantavirus from animals, and that's becoming, and that can create confusion in the anti-science movement. But it's quite clear that the importance of One Health, to think about health, not only at the humans, but at the environment and animals. Yeah. Just before I stop, we have two questions here. So that I can just read. Yeah. That's for Daniel Egan. Yeah, thinking about building capacity that can lead the African countries to less dependence on global supply chains. Yeah. He has strong memories on early days of the COVID pandemic. How much progress have you seen?
You worked in my group during COVID.
I
was working with me during COVID.
Okay. How much progress have you seen on this front in the past 5 years? Do you think we can achieve independence in this supply chain to safeguard the health of Africans? Yeah.
Great question.
It is a great question, and Professor Tobacco is very proud because you have worked with him. That asks very good questions. Yeah. So my answer it is that we have seen both. Sometimes we go 2 steps forward and we go a step backwards. Yeah. On relationship to the supplies, we see people thinking more about having supplies produced in South Africa and Africa. We ourselves are investing in companies on that, like Biotech. But sometimes it's also quite sad what happened with the mRNA. Yeah, it's such a great technology that don't know why they got bombarded by the anti-science movement, that we see some of the companies battling to take products to the market with higher accessibility. Yeah. Interesting, last week I was in Switzerland and we had a discussion with someone very high at Novartis, that when they say that sometimes people, they keep a lot of the things to be manufactured in Africa. But he said that sometimes the best thing that one can do in South Africa and Africa is not even the manufacturing, it's the R&D. We have such strong universities, and that sometimes we may think about a different, why we cannot do R&D for big companies that then can be, because commercialized, can be built, for example, it's very difficult to South Africa to be able to beat what Brazil and India have done, and China and South Korea in the manufacturing. Yeah. And sometimes what we have to think is what is our edge. And one of our edge is that South Africa really punch above the weight, on the science, and especially medical science. And one of the reasons it is to have a very well functioning medical research council and scientific community. Yeah.
If I can make a comment on that answer, in the early days of COVID, I remember, one of the response, talking to some of the big funders, we would say to them, if you really want to be helpful, charter airplanes and fly reagents. And some of the big funders thought we were joking, that this wasn't serious. And so has that change happened now? Because with Ebola, probably not. No. People still don't take it that seriously. And so until we start making some of these key things here, so some things have improved, but a lot hasn't.
Yeah. So just two quick questions, and then we can close down. Thank you for highlighting the importance of science communication. That's, by goodness, help me to read this name.
Aqua,
Mama, Ekua, Akona, Serlon. Someone that has a long name as longer as mine. Yeah. So yeah, thank you for highlighting the importance from Ghana. Okay, thank you. The land of freedom. Yeah. Thank you for highlighting the importance of science communication. Would you like more scientists to take up this skill? Yes, absolutely. Yes. Yeah. And last week, we also hosted the International Science Communication Conference. We did a pre-conference at SAMRC, which we funded. It's very, very important skill. But my suggestion is that people take that, but also take that very serious with good preparation, because some of the big media, the big media is good, but even sometimes the big media, so Al Jazeera, they can push you, yeah. And some of the fringe media can be very bad. They may look for the comma that you put wrong and do that. So it's very important training science communication, yeah. And that's something that we have to invest a lot more. I think it's a big gap. I do. Yeah.
Well, it remains for me to thank all of you for attending and engaging as meaningfully as you have, and to thank Professor de Oliveira and his team for the incredible contributions they are making, not only to our part of the world, but globally. Thanks to you.
Yeah, yeah.
Also, yeah.
I think that's all.